Let's recall. The active ingredient of traditional vaccines is the viral proteins, in one form or other, killed virus, live attenuated virus, or simply purified subunit spike protein. Remarkably, the COVID-19 pandemic brought about a genuine vaccine revolution, novel messenger RNA vaccines based on 3 technological innovations developed over decades. First, let's look at the target immunogen, the viral spike protein. Back in 2013, Barney Graham, working at the NIH on respiratory syncytial virus, showed that the trick for a successful vaccine was to stabilize the RSV spike protein by strategically mutating specific amino acids to proline. Proline tends to introduce local rigidity in the protein. In 2015, Barney applied this approach to MERS-CoV, introducing 2 consecutive prolines to stabilize the MERS spike protein. So, not unexpectedly, the moment that Youngjin Chung posted the CoV-2 genome on Twitter, Graham lost no time and within a month designed and then produced the DNA version of a CoV-2 stabilized spike ready for testing. Now, in parallel, a new technology was evolving, a technology that took over 20 years to get it right. The idea was to use RNA as the vaccine delivery system opposed to DNA or protein. RNA was hoped to be especially advantageous as it would enable the production of target proteins directly in the cytoplasm without the need for nuclear transcription, as would be the case for DNA-based vaccines. In order for RNA to work, it would need to comply with the format of native messenger RNA in our cells. Messenger RNAs contain an open reading frame, the actual message, which is flanked by 2 untranslated regions. The 5' UTR is preceded by a 5' cap structure and contains a ribosome binding site. The 3' UTR ends with a poly-A sequence. Producing synthetic messenger RNA was feasible. However, when synthetic messenger RNA was injected into mice, it caused a ferocious, out-of-control innate reaction. As you may recall, RNA is a potent PAMP, recognized by TLR3 and TLR7, which lead to an intense interferon reaction. Here is where Katie Carrico and Drew Weissman, working at the University of Pennsylvania, solved the problem. In 2005, they discovered that by replacing the uridine of RNA with a modified base, pseudouridine, our body would accept the foreign messenger RNA and not set off an alarm and safely translate the message into viral antigen. Thus, years before the emergence of CoV-2, Carrico and Weissman had already fine-tuned the use of messenger RNA as an efficient vaccine modality. Finally, the last problem was to deal with the fragility of RNA, how to deliver the RNA in such a fashion as to protect it from degrading and breaking down. Peter Cullies, working at the University of British Columbia, provided the answer. Cullies had been working for decades on liposomes, droplets of fat that can be used to package drugs and be injected into our bodies. The trick was to figure out the best combination that would be most effective in shuttling RNA into our cells. Cullies came up with a concoction of 3 lipids plus a unique ionizable lipid that together formed a lipid nanoparticle that could be used to package and deliver the messenger RNA. So, here we are, 3 technologies: antigen design and stabilization, RNA modification to enable messenger RNA vaccination, and lipid nanoparticles to package the RNA. Each technology the result of decades of research and perfection by hundreds of investigators, all come together as CoV-2 emerges. All you need now is to manufacture the vaccine and approve it for use. Fortunately, two relatively young companies were already active in developing RNA-based therapies and vaccines. BioNTech, established in 2008, and Moderna, founded in 2010. Both companies immediately developed lipid nanoparticle package messenger RNA, coding for stabilized CoV-2 spike protein, which in months after publication of the viral genome were already in phase 1 clinical trials. And by the fall of 2020, the results of phase 3 trials demonstrated efficacy of the vaccines. Moderna and BioNTech, who had partnered with Pfizer, had already begun production. The FDA in the United States gave emergency use approval of the vaccines, and people began to get jabbed as early as December 2020. Timing is sometimes everything. 3 technologies developed over decades were ripe and ready as COVID-2 emerged. And so, a new vaccine modality was designed, tested, manufactured, and deployed, all within 1 year.